Hi everybody, Professor Gustin here, and we're in microbiology, week 10. We're going to be talking about physical and chemical control of microorganisms, but we're also going to be taking lab practical too. So after you watch this video, what I would recommend is to do your studying for lab practical 2. Remember, it's the lab material from weeks 5, 6, 7, and 8. It's got 27 multiple choice ordering, multiple select, and short answer questions worth 50 points, and you have 70 minutes. If I want to give you an idea of what's on the lab practical, first of all, make sure you know the name of the tools that you used. And so I asked a couple of the tools. There's different things on colonies. What's a colony? What about media, the different kinds of media? We talked about selective and differential media. We used media like Sabouraud agar and EMB agar. You should know why we use those. What green metallic colonies mean on EMB agar. We grew things in fluid thioglycolate when we were looking at where the bacteria grew in the tube that told us about its oxygen requirements. So you should be able to interpret fluid thioglycolate tubes. We talked about colony morphology and mixed cultures and pure cultures. We did some things with selective and differential media. Remember, we did the dog print, and we were trying to figure out on the dog's foot, do we have soil bacteria, skin bacteria, or dust bacteria? So we did that by using selective and differential media. Other things that we did in lab, let's see. Yeah, colony morphology. Oh, the counting bacteria. I'll give you some real basic. If you have a good foundational knowledge of how do you count bacteria, counting the colonies, and how do you determine how many cells are in a milliliter of original culture or a gram of the original culture. So make sure you can do that. I give you a lot of time because I know there is a little bit of math, but I know some people get kind of panicky about math. So I made sure I gave you plenty of time on the practical. So study. Everything on the practical should be in your lab handouts. Use those completed lab handouts to do the lab practical. Also remember that we're using Respondus for the lab practical. So if you need to practice Respondus, I believe it was the Respondus practice was it in week 4? I think it was in the week 4. Where is it? Yep, there it is. The Respondus practice quiz was in week 4. So you can go to week 4 and practice that again to make sure that Respondus is working for you on your the computer that you're using. So we're doing a lot on physical and chemical control. Your book does a good job with that. And I talk it through using these videos. And you can complete your lecture study guide like you've been doing. And then all you have left after that is a lecture assignment. So we don't have any other lab assignments because the lab practical is taking up what we did, taking all our lab time. So the only assignment you have to hand in is your adaptive learning lecture assignment and your lab practical too. So I recommend doing the lab practical first because why fill your head with new stuff before you take a test on the old stuff? All right, you guys, good luck this week. I think this stuff is pretty straightforward, but if you want to talk about it, let me know. Give me a call, email me, whatever it is, we can talk. Hope you're doing well. Take care.